Cell biology is a scientific discipline that studies cells, their physiological properties, their structure, the organelles they contain, interactions with their environment, their life cycle, division and death. This is done both on a microscopic and molecular level. Cell biology research encompasses both the great diversity of single-celled organisms like bacteria and protozoa, as well as the many specialized cells in multicellular organisms such as humans, plants, and sponges. Knowing the components of cells and how cells work is fundamental to all biological sciences. Appreciating the similarities and differences between cell types is particularly important to the fields of cell and molecular biology as well as to biomedical fields such as cancer research and developmental biology. These fundamental similarities and differences provide a unifying theme, sometimes allowing the principles learned from studying one cell type to be extrapolated and generalized to other cell types. Therefore, research in cell biology is closely related to genetics, biochemistry, molecular biology, immunology, and developmental biology. Processes, movement of proteins. Each type of protein is usually sent to a particular part of the cell. An important part of cell biology is the investigation of molecular mechanisms by which proteins are moved to different places inside cells or secreted from cells. Most proteins are synthesized by ribosomes in the rough endoplasmic reticulum. Ribosomes contain the nucleic acid RNA, which assembles and joins amino acids to make proteins. They can be found alone or in groups within the cytoplasm as well as on the RER. This process is known as protein biosynthesis. Biosynthesis is an enzyme-catalyzed process in cells of living organisms by which substrates are converted to more complex products. Some proteins, such as those to be incorporated in membranes, are transported into the RER during synthesis. This process can be followed by transportation and processing in the Golgi apparatus. The Golgi apparatus is a large organelle that processes proteins and prepares them for use both inside and outside the cell. The Golgi apparatus is somewhat like a post office. It receives items, packages and labels them, and then sends them on to their destinations. From the Golgi, membrane proteins can move to the plasma membrane, to other subcellular compartments, or they can be secreted from the cell. The endoplasmic reticulum and Golgi can be thought of as the membrane protein synthesis compartment, and the membrane protein processing compartment, respectively. There is a semi-constant flux of proteins through these compartments. And Golgi resident proteins associate with other proteins but remain in their respective compartments. Other proteins flow through the and Golgi to the plasma membrane. Motor proteins transport membrane protein-containing vesicles along cytoskeletal tracks to distant parts of cells such as the axon terminals of neurons. Some proteins that are made in the cytoplasm contain structural features that target them for transport into mitochondria or the cell nucleus. Some mitochondrial proteins are made inside mitochondria and are coded for by mitochondrial DNA. In plants, chloroplasts also make some cell proteins. Extracellular and cell surface proteins destined to be degraded can move back into intracellular compartments upon being incorporated into endocytosed vesicles, some of which fuse with lysosomes where the proteins are broken down to their individual amino acids. The degradation of some membrane proteins begins while still at the cell surface when they are separated by secretases. Proteins that function in the cytoplasm are often degraded by proteasomes. Other cellular processes, active transport and passive transport, movement of the molecules into and out of cells. Autophagy, the process whereby cells eat their own internal components or microbial invaders. Adhesion, holding together cells and tissues. Division, by which cells reproduce either by mitosis or meiosis, cell movement, chemotaxis, contraction, cilia and flagella. Cell signaling, regulation of cell behavior by signals from outside. DNA repair, cell death and cell senescence. Metabolism, glycolysis, respiration, photosynthesis, and chemosynthesis. Transcription and mRNA splicing, gene expression. Internal cellular structures, chloroplast, key organelle for photosynthesis, cell wall, extra layer of protection, cell membrane, the part of the cell which separates the cells from the outside environment and protects the cell, as well as regulating what goes in and out of the cell, cilium, motile structure of eukaryotes having a cytoskeleton, the axonome. Cytoplasm, contents of the main fluid-filled space inside cells. Any chemical reactions also happen in here. Cytoskeleton, protein filaments inside cells, endoplasmic reticulum, major site of membrane protein synthesis, flagellum, motile structure of bacteria, archaea and eukaryotes, Golgi apparatus, site of protein glycosylation in the endomembrane system, lipid bilayer, fundamental organizational structure of cell membranes, lysosome, breakdown cellular waste products and debris into simple compounds, membrane lipid and protein barrier, mitochondrion, major energy-producing organelle by releasing it in the form of ATP, nucleus, holds most of the DNA of eukaryotic cells and controls all cellular activities, organelle, term used for major subcellular structures, ribosome, RNA and protein complex required for protein synthesis in cells. Vesicle, small membrane-bounded spheres inside cells, techniques used to study cells. Cells may be observed under the microscope, using several different techniques. These include optical microscopy, transmission electron microscopy, scanning electron microscopy, fluorescence microscopy, and confocal microscopy. There are several different methods used in the study of cells. Cell culture is the basic technique of growing cells in a laboratory independent of an organism. Immunostaining, also known as immunohistochemistry, is a specialized histological method used to localize proteins in cells or tissue slices. Unlike regular histology, which uses stains to identify cells, cellular components or protein classes, immunostaining requires the reaction of an antibody directed against the protein of interest within the tissue or cell. Through the use of proper controls and published protocols, specificity of the antibody-antigen reaction can be achieved. Once this complex is formed, it is identified via either a tag attached directly to the antibody, or added in an additional technical step. Commonly used tags include fluorophores or enzymes. In the case of the former, detection of the location of the immunostained protein occurs via fluorescence microscopy. With an enzymatic tag, such as horseradish peroxidase, a chemical reaction is carried out that results in a dark color in the location of the protein of interest. This darkened pattern is then detected using light microscopy. Computational genomics is used to find patterns in genomic information. DNA microarrays identify changes in transcript levels between different experimental conditions. Gene knockdown mutates a selected gene. In situ hybridization shows which cells are expressing a particular RNA transcript. PCR can be used to determine how many copies of a gene are present in a cell. Transfection introduces a new gene into a cell, usually an expression construct. Purification of cells and their parts. Purification may be performed using the following methods: cell fractionation, release of cellular organelles by disruption of cells. Separation of different organelles by centrifugation. Flow cytometry, immunoprecipitation, proteins extracted from cell membranes by detergents and salts or other kinds of chemicals. Notable cell biologists, see also. Cell disruption, cellular microbiology, Institute of Molecular and Cell Biology, Prokaryotic Cell, the American Society for Cell Biology, Cell Physiology, Notes, Penayahan, James E. Chapter 2. Technologies for detecting metals in single cells. Section 4. Intrinsic X-ray fluorescence. Inban C., Lucia. Metalomics and the cell. Metal ions in life sciences 12. Springer Doi, 10.1007-974-007-5561-12. ISBN A978-94-007-5560-4 Electronic book ISBN 978-94-007-5560-4 5561-1 ISNA 1559-0836 Electronic ISNA 1868-0402, Cell and Molecular Biology by Karp 5th ed., ISBN 0-471-46580-1. This article incorporates a public domain material from the NCBI document Science Primer. External links: Cell-Centered Database, Cell Biology at DMOZ, Aging Cell, Francis Harry Compton Crick by A. Andre at the Embryo Project Encyclopedia.